Good morning, viewer. Welcome to our FT English News breaking news. Lab-grown mini spinal cord. Amazing. A lab-grown mini spinal cord just showed that photostics dancing molecules might help heal paralysis. For the same, researchers at Northwestern University have created the most sophisticated lab-grown model of human spinal cord injury so far. In the new study, scientists worked with human spinal cord organoids grown from stem cells, and these miniature, simplified versions of the spinal cord allowed the team to recreate different forms of spinal cord trauma and evaluate a promising regenerative treatment too. For the first time, the organoids closely reproduced the major features of spinal cord injuries seen in people that included widespread cell death, inflammation, and a glial scarring. Viewers, glial scars are thick clusters of scar tissue that form after injury and create both physical and chemical barriers that block nerve repair. When the damaged organoids were treated with dancing molecules, a theory that previously reversed paralysis and repaired tissue in animal studies, and the result was striking. The injured tissue produced significant neurite outgrowth, meaning the long extensions of neurons began growing again, and skull-like tissue was greatly reduced. The findings strengthen the hopes that the treatment of which recently received orphan drug designations from US Food and Drug Administration could improve recovery for people living with spinal cord injuries. The study, published in the journal Nature Biomedical Engineering. Viewers, one of the most exciting aspects of organoids is that we can use them to test new therapies in human tissues, said Northwestern's Samuel I. Stubb, the study's senior author and inventor of dancing molecules. Short of a clinical trial, it is the only way you can achieve this objective. So we decided to develop two different injury modules in human spinal cord organoid and test our therapy to see if the results resembled what we previously saw in the animal model. So applying our therapy, the glial scar faded significantly to become barely detectable, and we saw neurites growing resembling the axon regenerations we saw in animal, and this is validation that our therapy has a good chance of working in humans too. When the exclusive report.